Hello, everyone. Welcome to today's live webcast, Reusable Device Cleaning, Disinfection, and Sterilization Validations, Overview Key Design Considerations for Device Engineers. This is Sarah from CBI, and I'll be your moderator today. Today's webcast is being sponsored by Pacific BioLabs. PBL is a contract research organization offering testing and research support services to the pharmaceutical, biotech, and medical device industries. They specialize in preclinical studies in support of FDA and international regulatory submissions. PBL is AAA-LAC accredited and ISO 9001 and 13485 certified. Now for some important announcements. This webcast is designed to be interactive, therefore your participation is necessary. You can interact with the speakers during the Q&A session by simply typing your questions into the Submit Question box located below the viewing window. You can enlarge the slide window at any time by clicking on the Enlarge Slides button located below the presentation window. The slides will advance automatically throughout this event. If you experience any technical problems with viewing or hearing this product, please click on the Help button below the viewing window. Now I would like to introduce today's speakers. Presenting first is Diana Louie, microbiologist and study director at Pacific BioLabs. Diana graduated from the University of California at Riverside with a Bachelor's of Science degree in biology and has been working at Pacific BioLabs as a microbiologist since 2008. For the past 2 years, she has been a study director with a primary focus in designing and validating cleaning and disinfection processes for medical devices. And in that time, she has validated reuse processes for 23 devices ranging from surgical forceps to challenging complex devices such as endoscopes. Diana is also responsible for investigating new validation procedures and technologies and improving Pacific BioLabs' testing capabilities. Additionally, she is involved in biocompatibility testing of medical devices. Our second speaker is Dr. Maria Lilybeth Simiolano, microbiology team leader, medical devices at Pacific BioLabs. Beth is a licensed physician in the Philippines, having practiced medicine there for 11 years, including 7 as an ER physician. In the hospital and ER, Beth performed and assisted on a variety of minor and major surgeries. Beth also holds a bachelor's degree in microbiology and has worked as a microbiologist in the U.S. since 2006. In this capacity, she has performed a wide array of compendial testing, including device testing and device validations. She is the microbiology team leader for medical devices and also serves as a client account manager and works closely with clients to research and design special testing protocols and procedures. Also joining us today will be Aaron Burke, the Director of Business Development, and Tom Spalding, President of Pacific BioLabs. Thank you all for joining us today. Diana, why don't you start us off?
Hello everyone, I am Diana, and my work at Pacific BioLabs revolves highly around cleaning and disinfection validations of reusable medical devices. As some of you may know, launching a medical device into the market is not as easy as 1, 2, 3. Nor that it ever has been that easy, but there are so many things you need to consider and so much research and testing involved. And not only that, sometimes you have to jump hurdles just to get by each phase prior to FDA submission. Wouldn't you agree that if you had a better understanding of the system and each phase better from the beginning, all preceding phases would be a piece of cake? Well, that is where we come in today to help you better understand reprocessing validations and how early design considerations with relation to cleaning of your device will help you get through this phase quick and painless. We will clarify what cleaning, disinfection, and sterilization validations are and the different categories of reusable medical devices with association of their acceptance criteria. Then we will reiterate and stimulate your minds with a few case studies that we have put together for you. And finally, we will go over some important design considerations for a reprocessing-friendly device. There will be time at the end of the webinar to discuss any questions you may have for us. Hold on, I have some technical difficulties with the slides. Okay, some of the goals we have for you today is to get a more thorough understanding of what a cleaning, disinfection, and sterilization validation is. At the end of the webinar, we want everyone to take note on the different design considerations there are and how you can increase the suitability of your device for reprocessing and reuse. Considering the designing of your device early on for cleanability will save you time and money. Before we get started, I would like to get an idea of what phase of designing everyone is at. Please let me know if you are at the early stage of designing a device and need more information on how to make it more reprocessing friendly, or if you have already designed a device and require its reprocessing procedure validated, or if the device is already in the market but FDA is requiring that you validate its reprocessing procedure, or you're simply looking for more information. Okay, so now that I have a better idea of where everyone is at, I want to start by discussing the basics. What is the purpose of having a reprocessing procedure validated? It's to set standards and prove that reprocessing procedure will render a device safe for patient use. And how does the FDA define cleaning, disinfection, and sterilization of a device? According to the FDA, cleaning is the physical removal of organic soil from a device to the extent for further processing or for intended patient use. Disinfection destroys pathogens and other microorganisms by physical or chemical means. And sterilization renders a product free from any viable microorganisms. But how are we supposed to go about validating this procedure? And how do we know it's validated? Well, let's break it down. First, what is a cleaning validation? As a reminder, cleaning is defined as the removal of soil residues, whether it be organic, inorganic, or biological. And how we go about validating a cleaning procedure is by first inoculating the device with representative organic soils and microorganisms at the most challenging locations to simulate the worst-case scenario. Organic soil may consist of anything that would be contaminated on the device during clinical use, and same goes for the inoculation of microorganisms. We inoculate microorganisms representative of the intended environment of your device. After introduction of soils, we subject the device to the recommended cleaning procedure. This procedure usually involves cleaning with water and detergents, but the recommended procedure can be suggested by the chemical manufacturer or established between the sponsor and the consulting lab. After we have cleaned the devices, we extract and quantitate residuals remaining from the cleaned devices. There are a few ways we can go about recovering the residuals, which can include a microbial test, also known as bioburden, protein, hemoglobin, and etc. Based on the results from the recovery, we can determine whether the validation has passed. First, we inspect that the device is visually clean after the cleaning procedure. For those of you with long lumens or channels in your device, this visual inspection will include observance of clear liquid coming out of those channels or lumens. Since we inoculated with a known concentration of soil, we can calculate the reduction in soil by comparing the positive controls with the clean devices. For microorganisms, a minimum of a 3 log reduction must be met. A screening for residuals must prove to be below certain limits. In micrograms squared centimeters, protein must be less than 6.4, hemoglobin at less than 2.2, carbohydrate less than 1.8, and endotoxin less than 2.2. If your device meets these limits, the cleaning procedure is validated. Now that the cleaning procedure is validated, the device can be subjected to the next step in reprocessing, which can be disinfection or sterilization, depending on your device. But first, we can cover what a disinfection validation is. As a reminder, disinfection is the process that kills pathogenic and other microorganisms by physical or chemical means. In order to validate a disinfection procedure, we first inoculate the device with representative microbes and organic soil. Then we clean the devices with the previously validated cleaning procedure before subjecting the devices to a proposed disinfection procedure using disinfectants or thermal applications, generally with the use of water under 100 degrees Celsius. Afterwards, we recover and enumerate any microbes remaining on the device. Based on the microbial recovery of the devices, we can compare the positive controls with the clean devices to determine if a 6-log reduction of microbes is achieved. If the device is both visually clean and satisfies 6-log reduction, the disinfection procedure would be validated. For a side note, according to the AAMI guidelines, you may want to consider screening for harmful residuals remaining from your disinfectant and its potential risk to the patient. Obviously, if you apply thermal applications, a screening would not be necessary. As you can see, in disinfection, only a 6-log reduction of microbes is required, but some devices are required to be sterile. For devices which are required to be sterile, sterility is the only option after cleaning. Sterilization is a process that renders a product free from viable microorganisms and is measured by the sterility assurance level. This is the probability that a product is non-sterile. For example, an SAL of 10 to the minus 6 is a 1 in a million chance of a non-sterile event. The way we validate the sterilization procedure is as follows. First, we introduce biological indicators to the device, typically consisting of bacterial spores. We then subject the device to the recommended sterilization process, for example, autoclaving, and then we retrieve the biological indicators and perform a sterility test. The acceptance criteria for a sterilization validation is pretty straightforward. For a non-critical device, an SAL of 10 to the minus 3 is acceptable. For a critical device, a SAL of 10 to the minus 6 is required. I have just gone over quite a bit of material, and it may still be a little confusing, but simply put, to validate the reprocessing procedure, we have to contaminate the device, clean it, and disinfect or sterilize the device according to the procedure being validated. Then we have to check the level of contamination remaining on the device after the cleaning, disinfection, and sterilization procedure. If all checks are acceptable, the procedure will be validated for use. This whole validation procedure doesn't look so confusing now, right? This next table presents the different types of reprocessing procedures we have discussed. There are 2 methods of cleaning: manual, which is cleaning with or without use of brushes and specialized tools. If your device is complex and requires meticulous cleaning, you may want to consider designing specialized cleaning tools for your device. The second cleaning method is automated cleaning with the use of ultrasonic and medical washers. There are 4 classifications of disinfection: low, medium, high, and thermal disinfection. It's determined by how thoroughly the disinfectant kills microorganisms. In high-level disinfection, there is a potential to render the device sterile. If your device cannot be sterilized or disinfected with chemicals, another option is thermal disinfection, which is the use of high temperature under 100 degrees Celsius. The level is categorized by the microorganisms that the process kills or is effective against. The level of disinfection will also depend on the type of disinfectant you decide to use. Most chemical manufacturers categorize their chemicals as high, intermediate, or low-level disinfectants. There are 5 common types of sterilization: high and low temperature, chemical, gas, and radiation sterilization. Now that we have gone over the procedure, we have to figure out which end process we should use for our device. Will the device's reprocessing procedure end at the cleaning level, disinfection, or sterilization? We distinguish that by classifying our reusable medical device. Your device is classified as critical if it is introduced directly into the bloodstream or other sterile areas of the body. Examples include surgical instruments, needles, catheters, implants, and invasive endoscopes. Your device is classified as semi-critical if it only contacts intact mucous membranes of the body and does not penetrate the bloodstream or other sterile areas of the body. Examples include non-invasive, flexible, and rigid fiber optic endoscopes, aspirator tubes, and respiratory therapy equipment. And finally, your device is classified as non-critical if it only comes in contact with the intact skin. Examples include blood pressure cuffs, stethoscopes, and external sensors. Each device classification will have its own acceptance criteria. Critical devices must be sterile with a sterility assurance level of 10 to the minus 6. Semi-critical devices must be sterile with a sterility assurance level of 10 to the minus 6 or disinfected with a 6-log reduction of microbes. Non-critical devices do not need to be sterile and therefore are the most versatile when it comes to reprocessing. The reprocessing can vary from being cleaned, disinfected, or sterilized, and the acceptance criteria is nowhere as stringent as critical or semi-critical devices. It is important here to use good scientific judgment to determine the best reprocessing procedure for your device. Now, with a general idea of what a reprocessing validation is, let's tie everything together in 3 case studies we have put together for you. In case study number 1, we have surgical forceps, which are used for surgical procedures and have a high potential of contact with the bloodstream. Because of this, the forceps is categorized as a critical device, even though it is simple in complexity. And due to the high risk of this device, it must be cleaned and sterilized. Surgical forceps may be simple, but they can cause validation problems. While high-level disinfectants can render the forceps sterile, many chemicals are not compatible with the stainless steel. They can cause rust and corrosion. Another thing to point out are the uneven grooves on the tip and grip points of the forceps. These grooves can trap unacceptable levels of residuals or contaminants. So what we did first was clean the forceps with Alkanox and a sponge, because Alkanox is a detergent which is effective, widely used in hospitals, and more importantly, will not cause corrosion. The sponge helps remove residuals that may be trapped in the grooves of the forceps. Step 2 is very important. After cleaning in the Alkanox detergent, we rinse the forceps under running water. Not only will this help wash away loosened debris, it will also remove any remnants of the detergent. This is true of any chemicals that may be used on your device. If the rinsing is not adequate, residuals from the disinfectants can potentially be very harmful to your patient. Next, we screen the forceps for organic carbon, which could originate from either the soil or the detergent. The screening helped confirm that the residual levels are acceptable. The last step in reprocessing the forceps is to subject it to the steam sterilization. The use of autoclaves is favorable in the hospitals because they are readily available and they are highly effective in sterilization. Perhaps the greatest advantage of steam sterilization for the forceps is that it is quick, easy, and no chemicals are involved, which avoids any potential corrosion issues. After sterilization, the forceps were sterile and ready to use. What I have for you next in case study number 2 is a blood glucose meter. For some of you who may not be familiar with this type of device, there are usually 2 components. One component, which you can see on the left of the picture, is the lancing device, which pricks your finger. Once you get a drop of blood on a test strip, you would insert it into the larger device shown on the right, which will read your blood glucose level. For this validation, we did not validate the lancing device because this component is usually disposable. We only validated the cleaning of the meter. Although the meter does not prick your device, your pricked finger can still come in contact with the device. So this device is categorized as semi-critical, which means that this device needs to be cleaned and disinfected. The meter has buttons and electronics, so it is medium in complexity due to the fact that you have to take care in not getting any liquid into the device. Some of you may recall in our acceptance criteria summary, we said that semi-critical devices needed to be sterilized or disinfected. So why did we automatically choose disinfection over sterilization? Well, first of all, the glucose meter that we validated did not have sealed crevices or buttons, which meant that immersing this device in a cleaning solution would destroy the electronic components of the device. The buttons and crevices also pose a risk when cleaning because contaminants may become trapped in those areas. And we chose disinfection over sterilization because the electronic components will be destroyed by high heat or even thermal disinfection. And these devices also have plastic casings which will warp or even melt under high heat. So what we did with this meter was clean it with sandy cloth wipes, which are widely used in the hospitals. We screened it for TOC to confirm that the cleaning will remove unacceptable levels of residuals that may be trapped on crevices or buttons of the device. Then we disinfected the device with the same sandy cloth wipes, only this time we left the device wet with the wipes for 5 minutes. This renders the device disinfected without the use of high heat. And the use of wipes were also convenient and did not require immersion of the device. So you can see, with the first 2 case studies that I have gone over, each had its own limitations and each had a different procedure in reprocessing. I have just presented examples of a simple device to a moderate device. Now I would like to turn the attention to my colleague Beth, who will go over one of the more complicated devices we have encountered.
Thank you very much, Diana. After those 2 cases, I will be discussing the third case, which is a diagnostic device used to visually examine the interior of a hollow organ, which is the colon. This device is considered semi-critical because it usually comes in contact with intact mucosal surface. It is required to be cleaned and disinfected, if not close to being sterile. Designing an appropriate cleaning and disinfection process for a complex device like this will depend upon many considerations. The efficacy of the cleaning process can be affected by its complexity and limitations. Let us understand better what are these limitations. First is the complexity of the design. It is difficult to design a cleaning process that is both easy to perform and effective. The simpler the device, the easier to clean. More complex device needs more time to perform the cleaning process. This device is composed of metals, silicon, and glass, which are more susceptible to scratch and deformities. Some of these materials are not compatible with some cleaning agents, as their use can result in corrosion or release of toxic byproducts. The knobs and screws are potentially trouble spots for retaining high levels of residuals. Not all parts can be immersed in fluid. We have to be very extra careful to protect the delicate parts to prevent breakage of lens, camera, or destroy any electronic components. Repeated reprocessing can also affect the functionality of the device. So how do we clean this device? And how do we validate this procedure for repeated use and come up with the same cleanliness result? First, we need to identify the most challenging areas where we should concentrate on developing an appropriate procedure to clean them. Because of the limitations from the previous slides, we have designed a cleaning and disinfection process that is easy and doable. This device is cleaned using a lint-free soft sponge to avoid scratching. Wiping with a sponge and water will also help in the removal of visible contaminants. The long flexible tube is immersed into Sidex OPA while scrubbing it with a soft sponge. Partial immersion accommodates the limitations of the device, yet still allows high efficacy of the cleaning procedure. All parts that cannot be immersed in fluid were cleaned using the sponge and disinfectant. These are usually the parts that don't come in contact directly with the patient's mucosal surface. All the parts that were immersed in fluid were rinsed thoroughly in clean DI water to remove any residuals from the soil and chemicals used for cleaning. Since this is a semi-critical device, it needs to be disinfected or at least close to sterile. High temperature is not an option for this particular device. Hence, the only way to make it safe for reuse is to subject the device to disinfection process after the cleaning. The immersible part was soaked in Sidex for 12 minutes, which is the time indicated in the manufacturer's manual for instruction. However, the exposure time varies from one disinfectant to another. Then after soaking, thorough rinsing was performed to remove chemical residuals. And how do we know it is acceptable? We have to perform TOC testing to screen the device and ensure that any organic residuals are within acceptable limits. Alternatively, we can also do screening tests such as analytical testing or quantitative analysis. Proper cleaning and disinfection and adequate rinsing will surely render a medical device safe for reuse. If in the evaluation it passes all the acceptance criteria, then the process is validated. That is all I have for this case. I will move on to the next slides, which are about device design and what design factors are important to assure the efficacy of the cleaning and disinfection or sterilization processes. Why is design important in manufacturing reusable medical devices? Design is the most important aspect in manufacturing reusable medical devices because a medical device intended for reuse must be properly designed to ensure that it will perform safely and effectively throughout its useful life. It is now required to have a validated reprocessing instructions before a product can be labeled as reusable. We all know that a well-designed device can be thoroughly cleaned and effectively disinfected or sterilized, whereas a poorly designed device is difficult to clean and more likely to be damaged as compared to a well-designed product. There are 4 design aspects to consider in manufacturing reusable medical devices. We have material design, physical design, total system design, and misuse-related design considerations. Let us discuss this one at a time. Designing a device is not typically done by a single person. There are so many issues and complications that come with every design we think about, and all these issues can be addressed effectively if design development is conducted by manufacturers and engineers in consultation with users and professional organizations. Meaning, input should come from those who use the device and those that are responsible in reprocessing it, so that the device can address the needs of all parties. Let me discuss the material considerations first. Selection of materials to manufacture devices plays an important role in design process. It is important to consider its biocompatibility as well as its compatibility with the cleaning reagents and disinfectants. Materials selected should be stable in the presence of chemical and under expected processing conditions such as temperature, pressure, humidity, and other environmental conditions. Take note that the combination of the material and chemical reagents may potentially release a toxic byproducts which could be harmful to users. In this sense, it should be compatible with most of the cleaning and disinfection reagents commonly used in healthcare facilities, or it is readily available in the market for home uses. We need to consider also leaching of materials that can happen as a result of repeated use and processing. If any materials in a device can be adversely affected by the cleaning, disinfection, or sterilization process, we should determine the limit of its reusability. It is recommended to perform a degradation study or exhaustive exposure to determine how many times the device can be reusable. These materials should also be evaluated for their capacity to retain unacceptable levels of residuals. For example, porous materials. These are prone to residual retention as compared to non-porous materials. Smooth materials are preferred compared to rough surfaces for easy cleaning. Some materials are prone to scoring caused by the cleaning tools used. A good example of this is the metal ends of brushes that scratches the device surface. In effect, this can attribute to retention of residuals and tissue penetration, which makes cleaning more difficult. Metals, as compared to polymeric materials, are more susceptible to corrosion, especially if they come in contact with bleach and hydrogen peroxide. Metals with coatings can be adversely affected by ultrasonic cleaning and also mechanical cleaning. These coatings can leach out from the device and may affect the user. Whereas, polymers commonly react to chemicals, making them incompatible with some cleaning and disinfecting agents. It may result to discoloration or release of toxic byproducts as a result of the exposure. Let's move to the physical design. In physical design consideration, manufacturers, engineers should establish that the size, shape, and configuration will not impose any constraints in the ability to clean, to disinfect, and sterilize these devices. It poses a challenge to any healthcare personnel assigned to perform the cleaning process if they are confronted with the following design features, such as long, narrow internal channels with small openings, devices with multiple internal channels. These are considered very challenging because they are not freely accessible. These are the areas that are difficult to reach when cleaning is performed. A small opening is a very good site for corrosion because it allows an aggressive, strong solution to stagnate and possibly form a red rust around its narrow opening. One thing more, it may not permit a common brush to pass through and clean the interior surface of the device unless a special kind of brush is designed for that particular device. Rough, irregular, and discontinuous surfaces, presence of ridges, crevices, sharp angles, and joints are difficult to clean. These areas can trap or retain microorganisms and impurities such as patient's blood and other body fluids or the patient's tissues, which are good reservoir for microbial growth. The next thing to consider will be the ease of disassembly and reassembly. A multi-component device needs to be disassembled to allow thorough cleaning. We need to take into account the number and size of parts in a device that requires disassembly, and also the potential for incorrect reassembly by the users. It is important to consider the probability of losing parts or breaking the device, resulting to a non-functional device. As a consequence, the personnel performing the cleaning process will become reluctant to disassemble the device, ending up with a device that is not properly cleaned. We have to avoid or at least limit the use of additional agents in reassembly, such as adhesive chemicals or lubricants, which later become toxic or harmful to users. If a device cannot be disassembled but has inner channels to which patient's blood or other body fluids can gain access to, it is important to include an opening or a port that facilitates flushing through the inner channels. Brushing should also be taken into consideration during the designing process of the device. It is beneficial to design a brush appropriate for the device for ease of proper cleaning. When we say total system design considerations, we are looking at the whole systems and how reprocessing affects the overall device. The design elements such as the components, subassemblies, and the complete device itself should be evaluated, how it is affected by the stress imposed by cleaning, disinfection, and sterilization procedures, and other stresses from mechanical, electrical, and environmental source. All of these conditions should be evaluated in terms of any potential effect on the device compatibility with other devices in the intended operating system. For example, a stainless steel should not be mixed with aluminum, brass, copper, or chrome-plated devices, as it can cause electrochemical reaction. What are the effects of selected cleaning process on delicate instruments? Choosing the appropriate method in the processing depends on how it affects the device. For example, the use of ultrasonic washers. This method of cleaning is popular because of its efficacy in removing debris from hard-to-reach areas. It may be perfect to serve this purpose, but consider the fact that ultrasonic energy can also loosen tiny screws of delicate instruments and might destroy coatings, glues, and amalgam in the devices. We have to bear in mind that adequate reprocessing of reusable devices is vital to protecting patient safety. What about the effect of reprocessing? Will it have an effect when the cycle is repeated over and over again? How long can a device be reprocessed and still safe to use? That repeated processing of a device can produce toxic byproducts, may cause corrosion, scratches, and deformities. At this time, the device has reached its limitation. Because of this, we have to determine how many cycles can a device withstand. What about the functionality? One of the effects of reprocessing that we have to consider is the functionality of the device after repeated reuse and exposures. Can we still get the same performance in its intended use? This is another aspect that we need to prove. The areas of concern will be the electronics, the microchips, lens, and other delicate components installed within the device. Cleaning procedures should include extra precautions for delicate parts or components like this. So why do we have to consider misuse-related designs in manufacturing medical devices? Because it does not only affect the functionality of the device, but may also impose danger to users. It is important to anticipate and determine what harmful effect that can happen if any incorrect cleaning, disinfection, or sterilization process is used. With this in mind, we can include a margin of safety just in case it happens. What happens if improper cleaning is performed? Well, the device might be deformed, screws may become loose, it may result to scratches, and can lead to leaching of device materials. Likewise, if an incorrect disinfection or sterilization process is used, it may result in corrosion or a toxic byproducts might be released. It can also lead to other physical changes like discoloration and rust formation. Consequences of improper reassembly includes breakage of the device or loose small parts. This may cause damage to important equipment in use, and subsequently, loss of time to reprocess can happen. In order to prevent any harmful effect, manufacturers should include a warning in the instruction for use, especially if it is known that serious damage could occur related to the device misuse. A good example of this is an accidental immersion of powered equipment or devices. Not only does it affect the functionality of the device, but poses great danger to users as well. In summary, cleaning and disinfection or sterilization validation is a major part of FDA approval. It is required to have a validated reprocessing instruction for reuse prior to release of product in the market. However, for those products already out in the market, we're recalled to comply with the same requirement. Secondly, many design factors can positively or negatively impact validation. And lastly, understanding the effect of cleaning and disinfection or sterilization process will help in designing a device that can be easily validated and reprocessed. So that's the end of our webinar, and we would like to open up for a question and answer session.
So I'm looking at some of the questions here, and I have some good ones here. Somebody asked, why is the sponge used instead of the brush? And do hospitals prefer one over another? And this actually depends what your device is. If your device is susceptible to scratching, like such as some metals, just like what Beth said, then you wouldn't want to use a brush because overbrushing can cause pitting and scratches on the device. So it really depends what your device is. If it's more of a plastic, you might be able to get away with using a brush, but that's where you can validate it in the validation. Number 22, someone asked on slide number 25, how did you determine the time limit for the disinfectant? And did the device have to remain wet for the full 5 minutes? We determined the time limit because the chemical manufacturer actually lists the time for exposure on the device to render it clean or disinfected. And based on the validation, that would be your minimum. So if the manufacturer says 2 minutes, we would try to validate at 2 minutes, but if it doesn't pass for your device, we would have to up the time exposure of the disinfectant.
Okay, we have another question here. It says, if a validation procedure doesn't pass, what typically happens next? If it doesn't pass, we don't stop from there. We try to investigate the cause of the failure, and we usually reassess the device and find out if failure is attributed to the device design or selection of materials used. If failure is associated with the recommended cleaning, disinfection, or sterilization process, then the validation will be repeated using more stringent parameters. In order to avoid high probability of failures, we recommend that method validation and trial runs or R&D study will be performed prior to the actual validation to save time and money.
I have someone else who asked the question of how do you check for sterility?
To check the sterility of a device, we will need to perform sterility testing or sterility audit. So we usually do it in a clean room, and all the devices, it depends on what type of the device that you have. We will have to do a direct transfer to a media, and then we try to incubate them for 14 days and observe if there will be growth on all the bottles or on all the samples that we tested.
And someone else asked, is the 3 log reduction per test sample, or is it okay to use the average log reduction? So for instance, a 4 log for one device and a 2.9 log for another device would average out to be about a 3.5 log. And that would count as pass, right? or does it fail because of the 2.9 log? We actually, it can't be from the average log. It has to be per test sample. So out of 9 devices that we test, if either one of them fails, we have to investigate why, if it's because of lab error or because the device has an error or there's something difficult about the device. And someone else wants to know, how many samples will I need to send in for a project? We typically ask for 13, but we can test with a minimum of 7 devices. Another question is, what are some of the most common detergents used in the cleaning process for reusable medical devices? And this depends on your device. Generally, we use Alkanox because it's widely available in hospitals. But if your device is always exposed to blood or tissue, then we might recommend Turgizyme because it's an enzymatic detergent and it's good for that use. Let me see.
So while Diana sorts through the remainder of the questions, I want to assure everybody that we'll be sending emails to all participants, and we will send customized answers to any questions that we don't get to during the Q&A period. We will also select questions that do not have any confidential information in them, and we will post a Q&A list on our website sometime next week. So I'd encourage you to check back at pacificbiolabs.com, and if you want to see what other participants we're interested in that we don't cover in the Q&A session. Go ahead, Beth.
We have another question here. It says, how do you determine what type of disinfectants to use? Actually, it depends on what is the use of your devices. That is what we based from what disinfectant we're going to use to disinfect your product. Say, for example, if your device is non-critical device, then we will have to use a low level of disinfectant. Whereas if you are going to use or you are going to validate a semi-critical device, then it will go from the medium level to high level of disinfectants. But it also depends on the client what they want us to validate with their product, because we need to consider also the compatibility of the device to the disinfectant itself, because even if we choose one disinfectant and it is not compatible with the device that they want us to validate, then we cannot use that. I have another question here. It says, what are the regulatory standards for cleaning and disinfection validations? We usually use AMI TIR 12, AMI TIR 30, and AMI TIR
Standard 58.
58. So those are the 3 standards that we've been using for the validation. Some of these standards are also considered ISO. Most of them.
Okay. I have another question here that asks, what are the typical timelines for each validation? If you only have one device of interest that you want to validate, and there's just one cleaning procedure and one disinfection procedure that you want validated, our lead time is about 4 to 5 weeks for a non-GLP study. For a GLP study, it will be anywhere from 5 to 6 weeks.
I have another question here. It says, how do you determine which microorganisms to use for SAL rod reduction? It depends on what type of sterilization are you going to use. Say, for example, you want high heat or moist heat sterilization, then you will be using the most common microorganisms that is used is the Bacillus atrophius. And Bacillus is Sterothermophilus. And if you're going to use ETO sterilization, then Bacillus subtilis or Bacillus atrophius is the organisms that they use for the log reduction. So it depends. We have different microorganisms with different sterilization procedures.
I have another question here that asks, how many devices are used in a validation? Like I said earlier, we would like to ask for 13 devices, and if you can't provide that due to money producing the device or something like that, or you just don't have the time to produce it in time for whatever your deadline is, we can validate with a minimum of 7 devices.
I have another question here. It says, does the drying process matter when autoclaving? Should we be concerned with water droplets are left in our device? It depends on what is the immediate intended use for the device. If it is a flash cycle that you only want to sterilize your device and use it immediately, then we are not concerned with the drying process. But if you try to sterilize a device and keep it for a certain period of time, then drying is very important because we don't want any droplet that is left behind, because it can be a very good spot for the microorganisms to grow, or it can be a channel for the microorganisms to get inside the instrument or the device that you want to sterilize.
Okay, I have another question here. It's asking, is the acceptance criteria for cleaning validations the same for all classes of medical devices? And it is. It's in the slide for the 3 log reduction of microorganisms, or the residual limits for depending on what you're testing for, whether it's hemoglobin, protein, carbohydrate, or endotoxin. And someone else also asked, how do I select the sample size? And we usually test in triplet kits, so 3 cycles of triplicate devices. So that's why we ask for 13 devices. We would treat 1 as negative control, 3 as positive controls, and then 3 sets of 3 test articles. There are a lot of questions here that we would like to answer, but I believe we will email you back either individually, or we're going to try and post up the answers on our website later on this week or end of next week.
Okay, Beth and Diana, thank you very much for handling all these questions. It looks like we're getting to the end of those. We feel like we should be answering over the air, so to speak, or online. And so we will, as we say, send individual emails. If we didn't cover your question during the Q&A session, you'll also be receiving a summary email from the conference organizers that will have a link to the PowerPoint presentation. So if you want to go and check the details on any slides, you'll be able to do that after the fact. And again, do check back on our website next week. We'll put a list of the questions were asked that don't involve any confidential issues up there, so everyone can benefit from the good questions we've gotten from all of you. And thank you so much for participating in this webinar today.
Yes, and thank you, Beth and Diana, for those insightful answers. Unfortunately, we are out of time. So I want to thank the audience for their interesting questions, our speakers for their exciting presentation, and our sponsor, Pacific BioLabs, for making today's webcast a valuable experience. This webcast will be available for on-demand viewing through December of 2012. You will receive an email from CBI alerting you as to when this webcast will be available for replay. Please invite your colleagues who may have missed this live event by forwarding them the announcement. Thank you again for attending today's webcast. See you next time. Goodbye.